Hello everyone, welcome to this webinar as part of our Shimadzu Solution for Pharma series. I'm Brian Demansky, Strategic Collaboration Scientist at Shimadzu Scientific Instruments. I'll be your moderator and we will hear from Dr. Mridul Mandal about the recently implemented USP General Chapter 1469 on nitrosamine impurities. Before we start, I wanted to share a couple of notes for our viewers. The webinar console has a variety of items to help enhance your experience and interaction with us. In the screen, you'll see the following items. The slides will appear in the center of the console. Directly under the slides, you will see the related content list with clickable links relevant to the material being presented today. On the top left of the slides is the Ask Question widget. Please submit your questions during the presentation through this widget, and we will answer them during the Q&A session. Just below the Q&A box are survey questions that you may fill out anytime during or after the presentation. On the right are the moderator and speaker bios. You may expand the items here to learn more about us. Finally, at the bottom pane are the icons to bring up all these widgets in the case that they're minimized or hidden from your view. All right, let's get started with today's presentation. If you're just joining us, I'm Brian, your moderator. Today we'll be hearing from Dr. Mridul Mandal, and our topic is nitrosamine impurity analysis by USP 1469. Dr. Mandal currently works as the Marketing Manager for Pharma and Biopharma at Shimadzu Scientific Instruments and has more than 12 years of experience in both academic and industrial analytical research supporting pharmaceutical science. With that, I will hand it over to Dr. Mandal.
Thank you, Brian, for the great introduction. Again, the webinar entitled Analytical Solutions for Nitrosamines in response to USP 1469. In this webinar, we'll be discussing our analytical technologies and methods to detect the nitrosamine amounts in APIs and drug products as proposed in the USP General Chapter 1469 that should take for preventing unacceptable levels of nitrosamine impurities in pharmaceutical products. Here is the outline of today's webinar. First, I'll talk about a brief overview of nitrosamines and their analysis, genesis of nitrosamines, then source of nitrosamines, and how much would be the daily intake limit with your drugs without harming yourself, that are related to considering the required LOQs for the analytical methods. Furthermore, I'll show you some methods developed at Shimadzu that are in line with the USP General Chapter 1469, such as Procedure 1, LC, high-res mass spectrometry, Procedure 2, head-space GCMSMS triple quad, Procedure 3, LCMSMS triple quad, and Procedure 4, direct injection GCMSMS. Finally, I'll summarize the main takeaways from the webinar today. Any molecules that contain nitroso-functional group are considered nitrosamines. According to the International Agency for Research on Cancer, IARC, these molecules are probable carcinogens. The FDA and ICH also classify nitrosamines as potential mutagenic compounds. Various regulatory bodies, such as the FDA, have extended the list of APIs and drug products for testing nitrosamines. However, developing sensitive analytical methods for nitrosamine poses an immediate challenges due to trace amount which may be found in pharmaceutical products. And here, this is the structure of nitrosamines and how it can form. Let me give you a brief history of nitrosamines in drugs. Over 300 nitrosamines were tested in rodent cancer biopsies in the 1960s. More than 90% of them were deemed carcinogenic. EU regulators first became aware of nitrosamines in medicines in mid-2018 in Sartans. In July 2018, FDA announced voluntary recall of several medicines containing valsartans following detection of NDMA. In October 2018, FDA posted laboratory test results showing NDMA levels in recalled valsartan products followed by NDEA. In September 2019, nitrosamine impurity has been detected in batches of ranitidine. In December 2019, EMA confirmed that trace amount of NDMA from a small number of metformin-containing medicine outside the EU. In July 2021, FDA was alerting patients and healthcare professionals on nitrosamines in varenicline. Here, let me talk a little bit about the potential sources of nitrosamine. Any material used for the development, like gloves, can be the potential source. Anything used for the manufacturing, like glass bottles, can be the source. Solvent used for developing, manufacturing, and analyzing may contribute to produce nitrosamines. Excipient manufactured without proper control or in a contaminated facility can be the source. Nitrites are common nitrosating impurities for nitrosamines that have been reported in many excipients at ppm levels and may also be present as contaminants in solvents and other materials used for developing and manufacturing. Some nitrosamines may be occurred due to the storing condition, temperature, and environmental conditions. On the other hand, packaging material may be contributing to produce nitrosamines as well. Please allow me to explain target eloquent limits of analytical methods. Daily acceptable intake or AI limits are fixed, but the doses is drug dependent. Sensitivity required is dictated by maximum daily dose MDD and AI of each compound. Acceptable nitrosamine content, that is AI divided by MDD. For drug product with MDD of 880 mg per day and AI of NDEA is 26 nanogram per day. So let's convert from nanogram per day to ppm. So if you simply divide it 26.5 by 0.880, that is 880 milligram per day to gram per day. So that is 30 ppb NDEA. Exception may be needed depending on the maximum daily dose or if more than one nitrosamine is expected to be present. Such cases should be discussed with the relevant competent authorities. MDD can vary greatly from as little as 40 mg of drug substances per day to up to 1200 mg per day. This can have a major effect on testing protocol and the target for LOD and LOQ. For some drugs, reaching the 10% of maximum AI level can be accomplished with any of the procedures, which on the other hand, with high daily doses may require more sensitive detection to demonstrate safety. Here is the daily intake limit list. That is the nitrosamine list. That is NDMA, NMBA, NDEA, NEIPA, NMPA, and NDIPA. AI limit for this NDMA is 96 and NMBA also 96. For all other nitrosamines, it's 26.5 nanogram per day. Now let's consider a drug has 880 milligram per day, and as per the previous formula, if we convert to ppb, it will be for 96 100.09 and for 26.5 it will be 30 ppb. Now let's consider an extreme example, that is 40 milligram per day, which is typical for olmesartan. So now all 96 nanogram per day will be 2400 and all 26.5 nanogram per day will be 660 ppb. Now let's consider something in between, that is 300 milligram per day, which is typical for irbesartan. That means that 96 nanogram per day now will be 320 ppb. On the other hand, all 26.5 nanogram per day will be 88 ppb. That means you understand, if the drug dose increased, the method should be more sensitive. Now I am going to talk about procedure 1, that is HR-LCMS. HR-MS, high resolution mass spectrometry. Here is our recommended hardware and method. You can see our LCMS 9030 Q-top in the left side. Analysis target were the same with the USP as shown here. Sample diluent was methanol. Mobile phases were 0.1% formic acid in methanol and 0.1% formic acid in water. Recommended system is Nexera USP-LC with LCMS 9030, which has very high mass resolution, very high mass accuracy, very fast acquisition rate. The mass range for quadrupole is 10 to 2000 mOV. The mass range for top is 10 to 40,000 mOV. Here is the results for nitrosamines in Metformin and Sartan drugs. A targeted MSMS top method was established for detection and quantitation of up to 8 nitrosamines using LCMS 9030. In the left side, you can see the chromatograms like NDMA, NDEA, all others, with separating from each other with the time within 15 minutes. And in the middle, you can see the analytical curves, that is 9-point calibration curve starting from 0.5 nanogram per ml to 100 nanogram per ml. And on the right, you can see that the chromatograms, which is from LOQ of the compounds. So the picture is still very nice shape and with a nice white and resolutions. Here is the complete results. So the rectangle box are showing these 2 additional compounds were analyzed with the USP and FDA recommended analysis of the interest. So that is the NDMA, NMBA, NDEA, NEIPA, NDIPA, and NMBA. In all 6 molecules, including other 2 as well, you can see that the LOD, LOQ range accuracy for like a LOQ 1 nanogram per ml, 5 nanogram per ml, 50 nanogram per ml, and also recovery, everything was done comprehensively. And this result shows that this method is very powerful to analyze the compound recommended by the USP. Chromatographic separation and identification of DMF and NDMA are essential to prevent the overestimation of NDMA when DMF interfaces. Here is another reason you can consider Q-top. According to a citizen petition filed by a private testing lab to the FDA, 16 out of 38 Metformin products tested had levels of NDMA exceeding the acceptable intake of 96 nanogram per day. In this case, an isotopic peak associated with an allowed DMF impurities was shown to impact the method only when insufficient mass tolerance was used. In order to ensure adequate assessment of nitrosamine levels in drug products containing DMF or other potentially interfering substances, care should be taken to ensure the necessary specificity in analytical procedures. In the left side, you can see NDMA, DMF, DMF nitrogen 15, DMF carbon 13 peaks. So let me explain you little bit in details. Insufficient mass resolutions or accuracy in data acquisition can pose the false calculation of NDMA. A mass tolerance window of plus minus 15 ppm or 30 ppm should be applied to obtain the EIC's presence of an interfering substances which correlated with NDMA. At 15 ppm mass accuracy, we observe 0.03% contribution based on intensity from carbon-13 isotope. We conclude that at the concentration DMF is present in these Metformin samples, there is no interference due to nitrogen-15 isotope of DMF. If DMF is correlating with NDMA, there may be some interference from the carbon-13 isotope of DMF, in this case about 0.03%. However, note that carbon-13 isotope can be easily distinguished from NDMA due to more than 115 ppm mass difference between carbon-13 DMF and NDMA. In our system, we can provide you with both on-column separations and mass accuracy. So that means NDMA and DMF were baseline separated with the chromatographic method. In the right panel you can see. And also there is a calibration curve which is 9 point. So no interference was observed from the nitrogen-15 isotope of DMF even at 15 nanogram per ml concentration of DMF in 10 ppm mass accuracy. Now I am going to talk about procedure 2, that is headspace GC-MS-MS. So what would be our hardware and method recommendation? So first, let me tell you the analysis targets as per the USP, that is NDMA, NDEA, NDIPA, and NEIPA. Sample diluent was methanol, carrier gas helium, and column USP G16. And this is our portfolio of GCMS. In the left side, you can see GCMS TQ8040NX with headspace 20NX, and then right side GCMS TQ8050NX with headspace HS20NX. If we compare with that you can still do the experiment with TQ8040 in the left side that has acceptable sensitivity, durable hardware, acceptable performance, reliable operation. On the other hand, right side, the sensitivity improved significantly and durable hardware, and the performance is superior and reliable operations. So if you need more sensitive instruments, our recommendation would be GCMS TQ8050NX with HS20NX. NDPA and NMPRG were additionally analyzed along with the USP recommended nitrosamines. Upper left corner you can see all nitrosamines are listed with the structure, and the upper right you can see the MRM transitions, and the red rectangles are showing the additional molecules. And the bottom NDMA analytical curves, and then overlapped chromatograms for those points used for the analytical curves. You can see there is very tight retention time, so peak didn't shift. And the right you can see a single chromatograms, and that is for the LOQ chromatograms, which has the very nice resolutions, shape, and width. All 7 nitrosamines were analyzed similarly and simultaneously. NMPRG was used and additional nitrosamines in conjunction with checking the quality of the processed water. Here are the complete results for all 7 nitrosamines analyzed by the headspace GC-MS-MS. When choosing the instrument for these procedures, be sure to consider how MDD of your drug compound will affect the required sensitivity for your analysis. USP 1469 proposed 20 ppb would be the acceptable LOQ. On the other hand, our developed method can be as low as 2.5 ppb. That means headspace GC-MS-MS with high sensitivity shielded detector provides outstanding noise reduction as well as excellent sensitivity, repeatability, and precision. Now I am going to talk about procedure 3, LC-MS-MS. What would be our hardware and method recommendation? In the left hand side, you can see Nexera USP-LC with LCMS 8045. Analysis targets were NDMA, NDEA, NDIPA, NEIPA, NMBA, and NDBA. Sample diluent was 1% formic acid in water. Deuterated nitrosamines were used for internal standard. Mobile phases were very basic. That means they were 0.1% formic acid in water, 0.1% formic acid in methanol, and the column was USP L1. Here is our portfolio of the LCMS triple quad. In this presentation, we'll show data for the LCMS 8045, varieties of detectors and source options to meet the analytical targets are available, and as needs change, the instruments can be field upgraded to the LCMS 8050 or even the most sensitive LCMS 8060NX. Prior to LCMS 8060NX, there is LCMS 8060. If you want to learn in detail, please visit our website or contact us. Here is the results for 6 nitrosamines in 5 Sartans. If you recall, the daily acceptable intake limits were NDMA 109, NMBA 109, all others were 30 ppb. In the right side, the table shows MRM transitions with the analysis of NDMA, NMBA, NEIPA, NDIPA, NDBA, and NDEA, calibration curve range, R square value, and LOQ. So we already achieved the very sensitive methods. As you know, sensitivity of the method plays a huge role if patients need high dose or the daily intake limit is higher. The method should be very sensitive. Here from the right table, you can see the method already very sensitive, so even a high dose drug can be analyzed by using this method. 9 levels of linearity was performed for all the 6 nitrosamines by using an internal standard. Here I am showing you only NDMA calibration curve and then overlay linearity standards. You can see that the peaks are very tight with the retention time. The peak didn't shift. On the other hand, if you see the LOQ chromatograms at the very right, that is the single peak. That's with the nice white resolutions and shape. That means this method can be used for very sensitive analysis of nitrosamines from drug products. Here is the results for the analysis of 15 nitrosamines. So you can see that the peaks are nicely separated and the list of nitrosamines are shown below. We have used LCMS 8045, and these instruments and methods can be used to analyze many nitrosamines at the same time simultaneously. Now I am going to talk about procedure 4, that is direct injection GC-MS-MS. Likewise, I want to talk first hardware and method recommendations. Analysis targets were NDMA, NDEA, NDIPA, NEIPA, and NDBA. internal standard was used, which was carbon-13 and deuterated. Carrier gas was helium, and column was USP G16. And here is our GCMS TQ8050 for direct injections method. A sensitive, reliable GCMS with direct injections has been developed for the analysis of 7 nitrosamines in Sartan products. The 7 nitrosamine analyzed include nitrosamines NDMA, NDEA, NDBA listed by both FDA and USP Proposed General Chapter 1469. The method displayed here highly sensitive and acceptable results with high SN ratios and low percentage RSD values. Here I am going to show you NDMA and NDA as a model. You can see that very nice calibration curve were made, and NDMA and NDA both 5 nanogram per ml peak. You can see that they are very nice shape, resolutions, and weight. As this is the complete results for the study, and this work was done prior to finalizations of the USP General Chapter 1469. We additionally analyzed NMEA, NDPA, NPYR, NPP that can certainly represent USP recommended nitrosamines. Here we will find solutions for nitrosamine analysis. Please click solutionsforpharma.com, then click genotoxic / impurity analysis. You will see a subpage, nitrosamine analysis in pharmaceuticals. This is our complete solution for nitrosamine analysis, and you can find columns, vials, solvents, reagents, and instruments like LCMS 9030 Q-top LCMS system, which is our high-res instruments, portfolio for triple quad GCMS with and without headspace, and very sensitive triple quad system like LCMS 8060NX LCMSMS system. What are the take-home messages from the webinars today? I hope you are convinced that analyzing nitrosamine impurities at trace levels poses many challenges which can be met with these workflows and instrumentations described here. Selection of instruments is dependent upon method goals, formulations, and a variety of other factors. But I can tell you, Shimadzu's highly sensitive MS technologies powered by analytical intelligence can identify trace amount of nitrosamine impurities in drug substances, drug products, excipients, and solvents. If you have any questions, even after the presentation, please send me an email, and my email address is mkmandal@shimadzu.com, which is shown here. Thank you very much for your kind attention. Now I am handing over to Brian Dominowski for leading the Q&A session.
So let's thank Dr. Mandel for such an engaging presentation. Again, under the related content list, you can find more information about our analytical solutions, including the nitrosamine analysis brochure, which is a brief compendium of the methods and instrument systems that we've described today, as well as more detailed application notes on each of the individual types of analysis. If you've not yet had the chance, feel free to fill out the survey questions on the left-hand side to provide us feedback and to request any additional information. We would also encourage you and your colleagues to sign up for the Shimadzu e-newsletter by responding to survey question number 3. This will provide you monthly newsletter information that has upcoming webinars and events that encompass a wide variety of application areas. We're going to go ahead and get started here momentarily with our questions. We've had some questions that have come in from the audience. Here's a good one. We have a number of different methods that have been presented. And in terms of using the QTOF and the triple quad methods for LCMS, what is the ionization source that has been used and why?
Good question indeed. We used both ionization sources, ESI and APCI, for the LCMS and found some nitrosamines are more ionizable with the APCI due to their nonpolar and volatile kind of structure. It is therefore sometimes recommended to use APCI over ESI if more sensitivity required for your analysis. Shimadzu has a dual ion source system capable of simultaneously doing the analysis or switching between ESI and APCI.
Excellent. Thank you for that answer. We have another question asking, how effective is sample extraction and recovery when the drug products contain complex formulations?
As the USP framework recommended that sample extraction can have a significant effect on sample recoveries. Therefore, Shimadzu tried that in advance. You are welcome to check the application note entitled Quantitation of 8 Nitrosamines in 12 different solvents by LCMS system that shows the extraction efficiency of different solvents on nitrosamines.
Excellent, thank you. I have another question that I see more than one user has asked related to where can we find a link for this webinar. I'll handle that one. You can use either the link that you used today to attend the live session. That will remain active as an on-demand link. And if you check soon to our ssi.shimadzu.com website or solutionsforpharma.com, you'll be able to find a link to the webinar in our video section on that website. That will be posted here shortly once we have today's session wrapped up. Let's return to another question. There's a question of, were standards used in terms of internal standards for this analysis for any of the 4 procedures presented?
Yeah, for procedures 2, 3, and 4, we use deuterated and isotopically labeled internal standard to check the accuracies and recoveries of the methods. But in QTOF, for method 1, which is high-res, we did not need any extensive or expensive internal standard as it's a high-resolution instrument.
Excellent, excellent, thank you. I see we have another question from a user asking if the maximum daily dosage that was referred to is based on API or total mass of the dosage form.
So I think it's an API, so the maximum dose they make as per the requirement of the disease. So the amount is shown here, that is the API, so that if you have more API, so that the formulation also increased, so that more excipients and other materials will be added. So at that time, you need more sensitive methods. But the methods we have shown here, that can go further down, so that sensitivity could be improved if you play with the method a little bit. So we can significantly increase your dosage limit as per the disease or as per the drug.
Excellent, excellent. Thank you for that information. I see there's another question. What is the impact for intermediate during oncology products regarding nitrosamines? I think the user is asking here about the danger and potential carcinogenic nature of the nitrosamine substances. And while I can't claim to be the expert on that, I'll field a little bit of an answer. At this point in time, the nitrosamine compounds that are of concern have been identified by ICH and FDA as potentially mutagenic substances. There's not necessarily a defined level that will relate to mutagenic states, but as you can see, there have been guidance levels set out for acceptable daily intake. So that's what we need to structure any measurements around, is that idea. I'm sure further studies over time will help to indicate the danger or potentially mitigate the perceived danger of these compounds over time, but I think it's wise that everyone is taking a cautious approach to this. Hopefully that helps to answer that question. We have another question, Riddle, that I'll field to you. So there's a question. Some of these are relatively in-depth. You may have to save the answers for those to offline. There's a question asking if method packages are available for LCMS for these nitrosamine compounds.
Yes, the method packages are available, and we can send. And in case you need to discuss with us, let's say you are developing an MRM-based method or direct injections or the method for the high-risk mass. So in that case, we can guide you to develop the method. So please feel free to contact us.
Excellent. Thank you for that answer. So we have another question about the LCMS triple quad system, asking if this is adequate for NDMA analysis in drinking water. Ultimately, I think that falls under an EPA guideline in the U.S.
Yes.
And at this point in time, I believe that LCMS is under investigation for the equivalent for the equivalent EPA methods. Those may have traditionally been GCMS or GCMS-MS method.
But it's possible. I would like to add a little bit, because EPA doesn't require that level of sensitivity like the pharmaceutical company requires. So in that case, I think it's very easily can be done. So instead of using ESI, you can add FECI. So FECI, when it will heat the water will vaporize and the sample will go directly to the mass spec. So you will be very easily to do that work. So if you need more information, please shoot me an email.
Excellent, excellent. And unfortunately, it looks like we've kind of hit the end of our allotted time for today for questions. I do want to thank everyone again for their attendance and let you know that we can find additional resources relating to today's topic at solutions.pharma.com. And we encourage you and your colleagues to attend our upcoming webinars. We will have registration information forthcoming, and check to solutionsforpharma.com for those upcoming events, or again, sign up for our e-newsletter. Thanks again to Dr. Mandel and everyone on the pharma team at Shimadzu for today's presentation.